Hi, everyone. I'm Kathy Campbell. I'm talking to you live from Auckland, New Zealand. All right, everybody. We just got ourselves sorted out here on Skype. So I just wanted to make a few introductory remarks and then open up for questions. As I mentioned, I'm Kathy Campbell. I'm a professor at the University of Auckland in the School of Environment. I'm a geologist, a paleontologist, a sedimentologist, and an astrobiologist. I've been working on astrobiology, exobiology topics for many years now, and just want to tell you a little bit about me and then what I do, and a few things about the academic path I'm on and other topics along those lines. So I studied in the U.S. You can hear my accent. I've been in New Zealand 18 years, but in fact I was trained at the University of California, Santa Cruz for my bachelor's, and that's when I discovered earth science. And did lots of other sciences in conjunction. And then I went on to master's at the University of Washington, where I also worked in geology as a paleoecologist. And it was there that I made some important discoveries that led me into exobiology. Then I went on to USC, that's the University of Southern California in Los Angeles, where I did my PhD work and worked then fully on extreme environment topics, which I discovered by accident during my master's. After that, I did a postdoc at NASA Ames Research Center at Moffett Field, California, where I became fully involved in my work linking to exobiology topics, which was what it was called at the time. And did some very nice work there and discovered hot springs. I had been working on hydrocarbon seeps, which is another type of extreme environment. So I found myself working on extreme environments as analogues for early life studies and also life on other planets. And it was because of NASA Ames that I got my job in New Zealand and moved to New Zealand in 1997. And New Zealand, if you haven't ever been here, is a beautiful place, and Joy knows. And there are many, many extreme environments. And I began working on them here, extended my work into South America, and then more recently started working directly on early life comparisons of hydrothermal systems with some of the oldest fossils in the world in South Africa. And so the work that I do, in part, is really about studying analog environments to try to understand the conditions under which early life evolved, with perhaps that helping us have a better understanding of where to explore for possible habitats for life, particularly on Mars. So that's some of what I do, where I've come from. The academic path that I got on wasn't really accidental. I knew from a very early age that I wanted to be a scientist. I was asked in primary school what I wanted to do when I grow up, and I said I wanted to be a researcher. I remember the teacher looking at me in surprise and asking me if I knew what a researcher was. But luckily, we had National Geographics at home, and I knew very well what a researcher was and had been looking at researchers studying coral reefs in Australia, and I just thought it was the best thing ever. So when I was at college, one of the crossroads for me was definitely trying to decide whether I should do science. Physics and mathematics were not my forte. I was better at chemistry and biology and geology. And an economics friend of mine studying economics said, "What are you going to let a little bit of physics and math get in the way of you doing what you really want to do?" And I never looked back after that. And then I just followed my gut intuition about what I was best at within my field, and that brought me to astrobiology and exobiology. So it was very exciting and a lot of fun and a lot of hard work. And one of the things that I think about when I think about my work and I think about astrobiology now and how many people are interested in it, the one thing that I think is important is to get a strong foundation in a particular field, a particular area of science or certain areas of science. In my case, it was geology and chemistry and a little bit of biology. And I do think that's very important because what's so great about astrobiology is that it encompasses so many sciences and so many fields. But it's important to have a grounding, strong grounding in certain areas. Otherwise, I feel that you can only really skim the surface because it's such a huge field. And there are so many people you can collaborate with that it doesn't matter if your specialty is in one area and someone else's is in another area, because astrobiology is right at the boundary of different disciplines. The benefits and challenges for me of doing science is that because I'm an academic, I get to teach, which I love. I get to do research, which I love. I get to go all around the world, particularly because of astrobiology. You go to the places where the exciting things are happening. So that part I really think is fantastic. Challenges are just the economics of the world that make it hard to do science. It's expensive sometimes, and sometimes it's hard to get funding. And sometimes universities are running on business models, which is a little bit hard when you're a scholar. But I think the opportunities are out there. I was a scholar for a whole year in France last year, working with people who are in the ESA program, which is the European Space Agency program, and also looking at the Archean or early life rocks in South Africa. So I managed to do that even on a shoestring budget. So those are really the only challenges, because my work is really a hobby. And I love going to work every day, and it's different every day. And I love working with students, and a lot of them are very excited about this type of subject area. So all of that part is great. When I also think back on early career, I guess the most important thing was following what I felt was my passion, finding my passion, then following it, not being afraid to follow it. And then when I was out doing my field work, really looking and seeing and making observations. And when I noticed something different in the field, something was strange about the rocks I was studying, that was the key to my whole career. And in the case of what I discovered, it was discovering some of the first fossilized hydrocarbon seeps in the world, which turn out to be another form of extreme environments that are just cold springs on the seafloor with organisms living around methane, with some implications certainly for exploring for life on the icy moons. But at the time, no one even knew anything about them. And luckily, I saw that there was something strange, and I started looking into it. And I went to zoologists, and I went to chemists, and eventually realized what I've been looking at. So my advice is to follow your gut, follow what you're good at, because in geology, I'm good at certain things, and that turned out to be what I do and what I'm very good at. So, and that began with literally finding some fossils that didn't make any sense, and making sure that I found out more about what they might mean. So that's the important thing. And then I certainly wish I had done more of the allied sciences. I did some, but I think it's worth the extra time when you're early in your career to really get a strong foundation in those allied sciences. I would have loved to have done more chemistry, for example. In terms of women in science, I've had a really good ride. I've done really well with that. My department is 50% women, and I find it incredibly interesting working both with men and women, students and academics. And so I'm very lucky that I'm in a program where I don't feel any kind of discrimination. It's hard work being an academic, and that's hard work for anyone, whether you're female or male. So I've just really, being a female in science is really exciting, and I just wouldn't let anything stop me from doing it. And it didn't ever get in my way. I had 4 male mentors who were older, and they were wonderful to me and helping me in my early career. So I found mentors, and I found study buddies. So I worked a lot with people to help me. And then in terms of astrobiology itself, that's an interesting question, really, about why is astrobiology cool. I found out about astrobiology kind of by accident. I started working on sedimentary rocks and fossils, and I wanted to understand the interactions between the sediments, the environment, and the organisms that were living there. And I didn't know I was going to study extreme environments. I just discovered this by accident. That really led me on to NASA, which then it became quite deliberate once I was at NASA, and I began to work on other extreme environments. So I would say that, it was exobiology then. I don't think it had the broad umbrella coverage that it has now. And yet everybody was excited about it when we were working on the projects we were working on. The Mars rock story opened up, and when I was at NASA, and it was very exciting when people were talking about the meteorite ALH 84001. That was all happening when I was there. I was very involved with giving away funding for that, studying that meteorite at the time as a young postgraduate. So why is it cool? Because we can really go to the boundaries of science, and we can really integrate sciences. It's not just me doing a bit of geology somewhere, but me trying to understand global phenomena that are about extreme environments and the significance of the evolution of extreme strategies for making a living. And you have to do that in an interdisciplinary manner, and I think it's really exciting. And of course, the planetary exploration aspects are fantastic. And the more that we work together, the different scientists, and you can do that in astrobiology, it's not only okay to do it, it's encouraged. It's the only way to really find the answers to many of the questions. And that part, I think, is really exciting. Plus, you get to go all over the world and meet fantastic people, like I was able to do just this year in the New Zealand Spaceward Bound program, how I met Sanjoy. So I think on that note, I mean, I'm happy to answer questions or talk more about any of those aspects, but I'm happy to open this up then for questions if you guys have any. So I've got a question here. What is culturally different about science in New Zealand and the US? That's a great question. I learned a super important lesson when I moved to New Zealand, and that was that it's better to collaborate than compete. I was in a lab where competition was really intense, not so much in the lab, but there were very few jobs in the US for paleontologists, and we were encouraged not to talk to anyone about what we were doing except when we gave a talk at a conference. And we weren't really encouraged to collaborate. When I moved to New Zealand, the 4 older male New Zealand geologists who helped mentor me into being a young academic, a successful young academic, that they were all about collaboration, all about sharing of information, all about working together. And I realized that that was an incredibly important direction that I needed to go in. And I wasn't where I wasn't so encouraged to do that. And I think that's what's so special about astrobiology is there's so much collaboration. And I learned a very important lesson then. Yes. Have I looked at the Opaline Silica outcrops discovered by Spirit rover? And if so, what do you think has been found? Sure. I really love that work. Yes, I love that work. Thank you. Yes, that's really interesting. I have looked at it, of course, only based on the publications. I would love to look more closely at the data and at the additional material that probably didn't get into the papers. And what I believe is that absolutely with that much silica, 99% silica or whatever it is, a huge amount of silica, then it almost certainly had to have been formed by hydrothermal processes. I don't have any question about that. The actual opaline silica material to me doesn't look like alkaline hot springs. What it looks like is something that we've got in New Zealand, which are a combination of acid sulfate chloride springs. So they're a mixture, they're low pH, and they're a mixture of these 2 types of fluids. So end member fluids would be acid, and you don't get silica deposits, or the other end member are the alkali chloride ones, which you get very distinctive deposits, which I don't see any features in those Martian deposits that look like pure alkaline ones. So I would say they look like, to me, acid sulfate chloride, which produce thin veneers or relatively thin hot spring deposits compared to, say, an alkali chloride spring like the ones that you see all over Yellowstone. So yeah, I do think they're hot springs for sure, and I think they are siliceous hot spring deposits. So I'm really excited about that work. I quote it all the time in my work. Yeah, how did I get to work at NASA Ames? What a great question. I was a PhD student at USC. I was working on hydrocarbon seeps, and I ran into literally, this is really important for students. I went for more than a decade every single year to GSA. At that time, AGU wasn't so big. So I went to conferences every year, and that is incredibly important for networking and not something that New Zealand students do very much of, unfortunately. We're very far away, but in the US, it's super important. And I would say also here in New Zealand, but there isn't as much money for students to go to conferences. And I ran into Jack Farmer, who was at NASA Ames, who is now at ASU Tempe, Arizona. And Jack Farmer said to me, "Do you have any microbial fossils in those hydrocarbon seeps of yours?" And I went, "Yes, I do." And he said, "I'd like you to come do a postdoc with me. I'll help you write the application." And we wrote an NRC postdoc together. And that happened in the hallways of GSA. Ha. Wow, okay. So the question, that's a really good one. You guys are really on the ball. The fossilized microbial mats on Earth, similar to the morphologies found on the Curiosity rover, the work from Nora Nofke. Yep, I've seen that paper. I know it's a hypothesis paper. Well, Well, of course, as a geologist, what I really want to do is go and walk up to some of those outcrops there on Mars and have a good look at them. Because, sure, morphology is really exciting, it's interesting, but I've been burned by my morphology before myself. I've got a paper coming out on the highest temperature hot spring deposits, which are geyserite, which form around geysers. And I've written a big review paper on that. And we had some fossils in it, which is very exciting. That's kind of very new. But one of the morphologies we were sure were fossils. They were spherical things, and they were found in the micron scale. And after a lot of study of them, we finally concluded they were inorganic. And that's another paper I'd like to write. And I was convinced at first that they were microbial. And it took a lot of really creative thinking and some extra analyses to really figure out that, in fact, they were not. They were inorganic mimics. So my feeling is we need to have these hypotheses. We need to have people like Nora putting out these hypotheses, but there's absolutely no way we can be sure of anything. The fact that she thinks they're extensive and all over the place, I don't see that even in New Zealand. You have very specific conditions under which you get thick microbial mats forming or even thin microbial mats forming. And she's got lots and lots of examples from those photos. And my feeling is that probably a lot of them are going to turn out to be mimics. But clearly, in terms of sample return, we need to be thinking about places like that where we can get something to look at. So I don't know is the answer, but I've been burned myself. You have to be careful, and you really have to. You can't just look at pictures. Can I tell you a crazy fieldwork story? God, I have a lot of them. When I was a master's student, I thought that I was invincible and that I was going to live forever. I was, young. And so I did all my fieldwork alone, which I would never do now. And it was in the middle of my PhD when I was out in the middle of nowhere in California in some serious bush country full of poison oak and chaparral. And I was out on my own then, too. Having just gone through some very wild coastal experiences with wild dogs and bears and all kinds of things in Western Washington. And I thought I was fine. And then I went out and did all this field work looking for hydrocarbon seeps in California in really remote country. And I drove my little Honda through streams, didn't get stuck. And I don't know what I would have done. I met some crazy prospectors looking for gold who, I was worried for my life with some of these guys asking me about where I would find gold. And then I'm out, and this was all happening during my PhD. And I was out crawling underneath chaparral in the middle of nowhere, underneath the chaparral, out far, far away. And I thought, God, if anything happened to me out here, no one would ever find me. And I was dealing with rattlesnakes on a daily basis and a bunch of stuff. And finally, the penny dropped, as they say in New Zealand, and I realized that maybe I should do field work with other people. And from that day on, I never did field work alone again. And that's been a while now. But honestly, I had beautiful experiences being alone, and people still do field work alone. But at some point, I decided that I valued my life more than capturing another rock and another fossil. So I've seen beautiful places and had a lot of things, climbed up cliffs, cliffhangers and all of that. But now mainly I do field work with other people. Do I have a preference of where the 2020 rover should go? Wow. Well, I was in Europe last year, and I tell you, the Europeans are not too excited about having 2020 because there's ExoMars 2018, and I think they felt like that collaboration between NASA and ESA somehow disintegrated, and I don't know if they were super happy about that. So I may be involved with some of the landing site selection for the ESA project, but that hasn't kind of come to fruition as yet. Landing sites, well, if it were me, I got to be honest with you. I don't know about all the engineering technology of where you can land and where you can't land, but where I would want to land is where we think there are actual spring deposits. That's where I would want to land, not in some place where you have terrain that isn't interesting, to somebody like me who really wants to go to a place. So where you really might have springs. So localized, very focused areas, if possible, associated with volcanoes. So I'd like that. What instruments should be on it? Well, I really like instruments that are like little microscopes that can also analyze like something like Raman or near infrared, where you can, at the same time, you can get composition and microscopes, because every bit of my work it all starts with these external sort of macro scale observations, but the real clincher is always at the micro scale. Let's see. Okay, what is my take on looking at life preserved in travertine deposits? Okay, as analogues for life on Mars. Yes, I've worked on lots of travertine. I prefer siliceous sinter because of the preservation quality. You know, silica really preserves microbes exceptionally well, not just in hot springs of various ages, but most early life deposits that are really worth their weight in gold have been silicified. So even if they were carbonate peritidal settings, if they've been silicified, you've got a chance of actually recognizing microbial signatures. And if you can't look at signatures at the micron scale, it's really great looking at these macro scale features, but you also really preferably need this micron scale preservation. And my issue with travertines in general is that the carbonate just does not preserve the biological signals that well, only when they've been silicified for various geological reasons. So I'm always looking at this sort of rapid silicification or rapid mineralization. And even though travertine grows at incredible rates, what I see is that the organic material is not templated that well. Occasionally, yes, but usually during early silicification. What do I do for fun? Oh my gosh. Well, I have a 9-year-old who keeps me very busy, and we were just surfing in Indonesia, which is not really my cup of tea, but it was really fun being there with her. But we like to go bike riding. We had an amazing time in France because the French don't run over their bike riders. They actually have bike trails. So I love that. I'm a real garden fanatic. In New Zealand, here in Auckland, we are in it's a cross between temperate and subtropical. So you can grow almost anything here except pineapple. So we have, things like guavas and all kinds of beautiful palm trees and stuff like that. And I love all of that. But we also have a million weeds. So when I am gardening, I'm battling the weeds here because everything grows really well. So I love that. And I love travel. We've been many, many places in the world. My daughter's been to 35 countries. And we love the fact that we don't live in the U.S. because then we can get this world perspective that's harder to get in the U.S. unless maybe you're a scientist or you travel for business. Most Americans don't have that worldly view. And I've really enjoyed being somewhere else and seeing the world from this perspective. So love cooking, took cooking classes and learned how to cook Polynesian food. Love Mexican food. We have to cook our own here because it's not very good here. So those are some of the things I like to do. Balancing the academic life and being a parent is a great question. So here's the pros and the cons from my point of view. We waited to have children until older. And that was because we're both academics. We decided we wanted to get our careers going. We had a lot of travel we wanted to do, which we still do with our daughter. And so we did the career path, and I became a professor at a relatively early age. And in New Zealand, a professor is like a chair, which is even more than a professor in the U.S. So it's a massive thing to get a professorship here, which I just got this last year. But so we had Ella at late 30s, early 40, and that was great. But I'm a little tired sometimes because she's got so much energy. And of course, at that point, we weren't really able to have a second one because everything just got started so late. I don't regret it now because it's easy to pick her up and travel with her. But what we did was she always was a part of what we did. So she knows about geology. She comes on my field trips when I teach field camp. We often mix vacations with work. And we travel a lot, which is our it's a great thing when you live in the corner of the world to go other places. So the way we balanced it was that she did go to daycare. I didn't stay home for a year. I might have gone crazy if I did. I'm not that kind of a parent. So what we do is we often go on learning expeditions. When we went to Turkey, we learned a lot about Turkish culture before we went. And so we kind of make it part of our lives to learn, to do lifelong learning, and she's a part of that. We're a big part of her schooling. So she's just integrated in more maybe than other kids, but she's happy and does all kinds of sports here and is really happy in school. So I think we're doing okay so far. But yeah, I sometimes burn the candle at both ends. And now that I'm in academic leadership, I have that as well to do. So sometimes I just can't get everything done, and assignments are graded later than I would like, and things like that. And I've learned to let go. Learned to let go of being a perfectionist. I can't be a perfectionist anymore, which is probably a good thing. Okay, extracting DNA or lipids from travertines. Yeah, don't know about DNA. Lipids would be great. You know, I got to tell you the truth, and maybe you guys know something about this. I've tried for many years to get reliable biomarkers from sinters and travertines. And the problem that I've had is that I haven't been working closely with anyone. So my biomarker connection people have all had their own careers, their own interests, their own projects. And then I knock on their door and say, can you help us do some biomarkers? And we've run into issues with contamination. We've run into issues with material that is morphologically beautiful microfossils, terrible biomarkers. The silica just trashes them. And so when I work with biomarker people, which I've tried now twice, we sort of start on things and then it never really gets done, never gets finished because these guys have their own work. So I would need to have a much closer collaborator, I think, to really work on biomarkers, work on sampling properly for not having contamination, and also trying to find the right kind of samples, not the morphological samples that are perfect, because those turn out to be pretty much empty of biomarkers, but other things like maybe organic, rich, darker, low temperature deposits that seem to have more organic material preserved. But we have not been able to follow through with that because I haven't managed to get the right combination of collaborators, money, and rock samples all in the right place at the right time. So it's one of these things about my career that hasn't really come to fruition, even though I would love for it to. And what I found is that ramen microscopy has been the best thing. Mapping out carbon in situ, in thin sections, has been the best thing that we've been able to get the signatures and the textures in the same location. But unfortunately, so far, biomarkers hasn't been successful, but I think it could be. El Tatio. So I work on mainly, so I've been asked about the El Tatio centers. I haven't worked on them personally. They do have some mineralogy that, if I recall, is rather interesting. And I haven't worked on them because the Brian Jones's group, I believe, had done some work on their textures before. But perhaps these integrated microanalyses that we're starting to do, where we work on, we're using the Pixie microprobe in France, which is turning out, and laser ramen work is fantastic. And it might be that El Tatio could be useful for that. New Zealand, I work in New Zealand, and I work on Jurassic travertines and sinters in Patagonia, which are like a stepping stone into deep time, because a lot of them are turned to quartz already or have been certified. And so I'm finding the Jurassic to be the most useful analog for Archean work that I've found. And we've got a paper coming out in Geology on that pretty soon. I'll be looking at the Pilbara samples as well with respect to hot springs, but I haven't looked at El Tatio because other people have worked there, and we would need to have a new angle. But this new work that I've been doing, really integrating from the macro regional geology scale all the way down to the outcrop and then micron scale, using in situ techniques to really interrogate the stable isotopes, sulfur, carbon, and also looking at laser Raman and not yet biomarkers. That has been really powerful. And comparing our hot spring work then to the Archean, that's the work that's coming out in Geology soon. And sure, if there's something to look at in El Tatio, I'm there. If anyone wants to work there, great. Because I love working in Latin America, and I have a big program. What we're trying to get funded to do some really detailed work in Patagonia. So I'd love to. I've been near El Tatio, but I haven't actually been there myself. Yeah, outreach. Great question. How much fun. These questions are great. Okay, so outreach, I think, is really fundamental. I had not been focusing on outreach until quite recently, and luckily, with Spaceward Bound New Zealand that just happened in January, where I've met some amazing people. And also probably the launching of New Zealand Astrobiology, a network, if not an institute, with some other collaborators here is the way to go for us, because the high school teachers here now have to teach earth and space sciences in their curriculum. And a lot of them just aren't prepared and don't know how to do it, and they want to do astrobiology. So I stepped forward and said, okay, I will now start doing outreach in a much more integrated or systematic way. I'll put more of my energy into it. I think it's incredibly important that we do this. New Zealand is a little bit behind in this, in that the academics tend to not be linked up with the schools. But that's really not a difficult thing. They come to look at our volcanoes anyway, and all we need to do really is expand our networks from just looking at Auckland's volcanoes out into a larger brief. And that's something that I've started working on just this last year. Invite the teachers to join SagaNet. Yes, I think we will. My collaborator here, who's an astronomer and a passionate astrobiologist, and I are going to be this year trying to sort out the best ways to get people linked up and for us to find out what the teachers really need. So that's underway here, and I think it's very new to New Zealand, but I think it's the perfect place to work on it. We've got astrobiological topics and people working on those topics all over the place. We just need to link up the schools with like what happens with NASA, for example, or SagaNet. Biggest spot size. Yeah, wow. I would love to see an instrument on remote ramen detection. I think that is the way to go. If it were my instrument, that would be it. A microscope and ramen together are phenomenal. And I'd be more than happy if you want to email me. Once this paper is accepted, it's actually gone back to the editor with revisions. I'd really like to show you what you can do with beautiful analog samples. But the largest, let's see, the biggest spot size, we were doing maps that were, I'm just trying to remember the size of the maps, but I thought our maps were up to 1 millimeter by 1 millimeter. I know that doesn't sound very big, but there was heaps going on in some of our deposits. And I could actually find out what the largest spot size is from the guy we're working with in France, who is just a whiz with the micro ramen. So if you just want to email me, and we can take this offline and I can get you more information about that. But boy, that would be my instrument of choice, I can tell you that. We've just had the most fantastic results combining, once you understand your context, there's a huge amount of work going into the geological context, the environmental setting. Once you do that work, then you can do beautiful microanalysis and really prove the relationship between minerals, carbon, and microbes. It's just a really powerful technique. My favorite one. Yeah. The most awe-inspiring geological site I've ever been at, and why. The luckiest thing is my being able to go see fantastic geology all around the world, and that's a super hard question to answer because I've seen so many beautiful things. I've been so very lucky. But I'm a little bit biased. I have been dabbling and working on paleotsunamis for probably since the early 1990s. And I have a location looking at Pliocene ones, maybe even Miocene ones. There's about 3.5 million years old. And I think they are the best examples of tsunami deposits that are known in the fossil record, not by asteroid impacts, but by actual great subduction earthquakes. So the location is a remote cliff section in the Olympic Peninsula in western Washington. The place where there are all the bears and the cedar forest, and just spectacular. Salmon runs, the whole thing. And there are some unbelievably beautiful paleotsunami deposits. And what is so striking about not only seeing these deposits that were bearing estuarine marsh plants and just really spectacular, huge, thick things. What's so spectacular about it and so awe-inspiring is that as you stand on the beach looking at the sea cliff, you realize that you're standing in a great subduction zone right this minute, and that a tsunami, a great subduction earthquake and a tsunami could occur at any time. So that history goes back, 30 or 40 million years. And it's very awe-inspiring to be standing looking at fossil versions of these things and realizing you could be wiped out at any time if there's a great subduction earthquake. Because there's no way out. It takes an hour to get out of there, and et cetera, et cetera. So I found that to be most awe-inspiring. But there's lots of other ones. Yeah. the ramen work is going to be coming out in a geology publication. We just sent the revisions back to the editor, who said pretty much, yes, we want this paper. You just need to address some stuff. So I sent that out actually yesterday. And that's the culmination of my work working on South African Archean sediments that are full of fossils, microfossils, and both biofilms of photosynthetic organisms as well as chemotrophs that were living in the sediment. So a diverse microbial community that's 3.3 billion years old. And we did some beautiful laser ramen petrographic and pixie work, which is the compositional work where you can do maps at the scale of ramen and get all kinds of fine detail on quantitative elemental analysis, showing that there's a hydrothermal signature in the Archean, and then comparing that to our Jurassic hot springs. So that work is going to be coming out in geology whenever they put us in the queue and accept the revision, which I'm pretty sure they will. So that was a huge culmination of work in my career. I've always wanted to work on the Archean, and I finally was able to connect hydrothermal systems, hot springs, with Archean rocks, and hopefully it will be done also in the Pilbara, where I'm going there in June with Martin van Kranendonk to see what they're doing over there. So that's very exciting work. I'd be more than happy to send you stuff about that. I just need to wait maybe a few weeks just to make sure it's accepted. So just email me and I can send you that information. But the ramen work was fundamental. Where would I like to explore for life in the solar system? Well, I'm pretty boring. I mean, Mars, of course, for me. And it's because of the volcanoes and the sedimentary processes in the water. I'm very interested, though, also in the icy moons, but not so directly. I've got a Spanish collaborator here from the Astrobiology Center in Madrid, and she's here for this year and is looking at hydrates. We have hydrate deposits offshore in our subduction zone to the east of North Island, and she's going to be looking at data sets on hydrates and then comparing that to her planetary geology work and experimental work on hydrates. So I have a distant interest in hydrates because I work on hydrocarbon seeps. And I'm also interested in, of course, Martian exploration. But for me, I'd rather land on a big volcano on Mars and walk around looking for hot springs. So I don't think the rovers are doing that yet. How probable do I think that life exists in the solar system? I go back and forth on that. I have felt often that there is no life in the solar system, but I do think we have an obligation to look for it. I think the chances are much greater outside of the solar system. So that's much more remote, of course, but the exoplanet work is very exciting to me. Planets that are in the Goldilocks zone, planets that maybe they're Earth-sized. There's just too many planets out there for there not to have been life evolving more than once. I just can't logically understand why life wouldn't have evolved again if it evolved here. So I believe in life in the universe somewhere else. I'm not totally sure that our own solar system has got the signatures, but I would say Mars would be, for me, the best place to be looking. And I'm really keen on the subsurface because of all of the fantastic extreme environment analog work that people do here. I think the subsurface is a great idea, but how to explore for that on Mars is a big question mark. So in the meantime, hot springs are my favorite spot. Okay, last words of wisdom to early career scientists. Look, I think don't worry, be happy. Love what you do. If you don't love it, don't be afraid to change. I know at least two women I know who have recently changed careers abruptly, and in their 40s have gone and studied totally different areas. They're close friends of mine, totally different areas with 20-year-olds, and they're in their 40s, and they are so happy finally doing what they love. So don't worry if you don't find it right away, but really follow your heart, follow your intuition. Follow what feels great. Be interested, be creative, be passionate about what you do. And if what you're doing isn't so fun or interesting or creative, see if you can make it that way or find another direction. And it's okay to go in different directions. I've been kind of a classical academic. I've started in one area and mainly stayed in there, but just branched out. But I have seen people do very creative work and apply science in incredibly creative ways. All kinds of consultants I've met who do educational outreach in science, all kinds of things. So I'd say just the sky's the limit, but it's following your heart that's going to take you to where you want to go. That's for sure the case and happened for me and for pretty much everyone I know who loves what they do. Very happy to talk to you. Would be happy to do it anytime. It's been super fun. Your questions have been the most fun I've seen in a very long time. So thanks a lot.